far reaches of the American Southwest desert. He's dabbled in radio broadcasting, particularly the works of J.R.R. Tolkien. He's also dabbled in a mobile hot tub concept business entitled The Big Splash. And not just talking about starting a mobile hot tub business, as many of us may have at one point or another, but actually buying the hot tub and the trailer and getting into the repairs and maintenance of this hot tub. He got out of the business because the repairs were too challenging. He's also dabbled in international diplomacy by his accidental use of the diplomat lane at the Canadian border crossing. So, as I mentioned at the beginning, he's really brought a great skill set and broad perspective to this project, which I think was key to his success while undertaking this project. This project was funded by Sea Grant, as he'll mention as well. This has been a data synthesis project using a lot of our existing data here at SCDNR, as well as various publicly available data sets. And the goal was basically, in a broad sense, which he'll define better in just a minute, to explore how predicted changes to climate, you read all these climate reports and such, how those predicted changes may result in changes and impacts to our coastal system, specifically here in South Carolina. With regard to a technical aspect of the Zoom, I'm sure many of us are familiar with this platform by now. Questions, please hold your questions till the end. We will ask questions from the general audience and then excuse the general audience for committee questioning. I will moderate those questions as Lloyd's advisor. And if you have a question, please either type your question into the chat dialogue, or if you prefer to ask your question verbally, please type in the chat dialogue that you have a question and I'll call on you. And this will help keep things organized. And without further ado, let's get started. Let's get Mr. Lloyd queued up and take it away, Lloyd.
All right. Well, thanks for that wonderful introduction. Can't say, let's see, I need a screen share here. And all right, is this working?
Yeah, looks good.
Okay. So yeah, like Andrew said, this is a project we've been working on for the last 2 years. It's funded by the South Carolina Sea Grant. Been working with the environmental research section over at DNR. And the goal of this project is to explore the connections between land use, climate, and coastal ecosystems across the coast of South Carolina. So South Carolina's coastal landscape is really defined by its estuaries and tidal wetlands. Nearly 500,000 acres of salt marsh line the South Carolina coast. It's more than any other state on the Atlantic. These are highly productive and incredibly diverse ecosystems that provide a wealth of ecosystem services. They serve as nursery habitat for several important fishery species. Intact salt marsh vegetation is the first line of defense against rising seas, storm surge, and coastal erosion. And this natural landscape also adds important social, cultural, and recreational value to the region. However, estuaries and the coastal wetlands are threatened around the globe, and South Carolina is no exception. So, anyone that lives in the area knows South Carolina is a rapidly growing state. Much of this growth is happening within the coastal region. To accommodate this influx of new residents, we're seeing widespread shifts in land use and land cover. So in this graph here, these are the coastal population totals between 2001 and 2016, coastal South Carolina. And you can see during this time period, we grew from around 1 million to 1.4 million residents. And paralleling this growth, we see an increase in impervious surface cover from around 2.1% to 2.75%. So what does this mean for the estuarine ecosystems? Well, impervious surface cover can intensify stormwater runoff. So in a natural watershed, rainfall is intercepted by vegetation, slowed down, and allowed to percolate through the soil. But in an urbanized watershed, rainfall comes in contact with these impervious surfaces, such as pavement, rooftops, and roadways. This can exacerbate coastal flooding, which anyone that lives in Charleston is pretty familiar with. But additionally, the stormwater runoff carries with it several pollutants that make their way into the downstream estuarine systems. So I know the physical landscape is changing. What do we know about the climate that's changing? This graphic here is from the 4th National Climate Assessment Report, and it shows the anticipated increases in temperature, annual average temperature for the United States. And what we're seeing for the Southeast are increases of around 3.4 degrees Fahrenheit in the lowest emissions scenario by the mid-21st century, all the way up to 7.7 degrees Fahrenheit in the high emissions late 21st century scenario. So as the climate's becoming warmer, also expected to become wetter. So this graphic is from the same climate assessment report, saying for the Southeast, we're transitioning to a generally wetter climate, but there's still significant season to season and region to region variability. And what this means for the Southeast, that we might see the largest increases in precipitation during winter months. And while climate's becoming wetter, I think the nature of precipitation is also expected to change. So climate assessment report projected changes in extreme precipitation events. They define these as daily precipitation totals in excess of the 20-year recurrence interval value. And in their scenarios, expecting a 9% increase in these extreme precipitation events in the lowest scenario, all the way up to 21% in the high emissions scenario by the end of the 21st century. So previous studies in coastal South Carolina have linked the upland development to downstream degraded estuarine habitats. Some of these effects include higher levels of sediment contamination, higher concentrations of bacteria, as well as decreased abundances of the resident biological communities. Now, the goal of this project was to expand the scope of these earlier studies by expanding the geographical range to cover the entire coastline of South Carolina, as well as updating it with the most recent available data to see if these past trends continue into the present, all while adding an additional theme of climate change. And this leads us to the primary research question: how will the estuarine environment respond to a changing climate and changing physical landscape? So early on, when I was developing a thesis proposal, worked up these two primary hypotheses. The first, related to land use and land cover, states that degraded metrics of environmental quality in estuarine systems will relate to increasing development intensity within connected upland watersheds. These environmental impacts may include higher levels of sediment contamination, lower nectin diversity and abundance, fewer stress-sensitive benthic and vertebrate taxa, as well as higher frequencies of bacterial contamination. The second hypothesis related to weather and climate, where weather patterns associated with climate change, such as higher temperatures and extreme rainfall events, will be associated with these same degraded metrics of environmental quality. So to answer these questions, we employed a data synthesis approach. What this means is instead of collecting data on our own, we'll be using a variety of secondary data sources, connecting them across space and time to test our hypotheses. So we collected data on weather and climate, as well as land use and land cover. These made up our explanatory variables. And we link these with environmental data that describe the estuarine habitat. These are response variables. Through different statistical techniques, then crafted predictive models that could be used to predict the estuarine environmental response under different climate change and development scenarios. And ideally, these findings would go to inform policy and management strategies that could then mitigate some of these harmful effects. So this data synthesis methodology followed this sequence where we collected the data from a variety of sources, processed them so they were usable, integrated them, which is linking them across space and time, and made them ready for the data analysis. So for the physiographic data, these are data that describe land use and land cover. Our primary product was the National Land Cover Database, or NLCD. We have a few different products. One that we used was the NLCD land cover classes. So they break the landscape into 16 different categories of land cover. These include different types of forest, marsh, wetlands, as well as levels of development. And they also offer a layer of urban impervious cover. So this, and that's what's shown here on the right, the Charleston area. So this assigns each 30-meter pixel value a value from 1 to 100% based on the level of impervious cover. These are also available for multiple years, beginning in 2001 and as late as 2016. We got data from the most recent census on population, collected data on hydrologic soil groups. So these describe how well or poorly drained the soils are. And for stormwater infrastructure, we used DNR's stormwater pond inventory data set that tallied up the stormwater ponds across coastal South Carolina counties. These data were processed in GIS. Here we use several different spatial grouping systems. The primary one was HUC watersheds. These are USGS delineated watersheds for a standardized watershed grouping scheme of different sizes from 8 digits, which are the larger river basins, down to 14 digits, which are the smaller subwatersheds. We also had our own grouping system of grid cells where we overlaid the coast in different square grids of different sizes, and also analyzed data by circular buffers from 1 to 3 kilometer radius of each environmental data point. With these data, we could then calculate different land cover composition, the average impervious cover, as well as population density, and the count of stormwater ponds. And here on the right is an example of 14-digit HUC watersheds averaged by impervious cover. And here you can see the areas of Charleston and Myrtle Beach standing out, with also some more rural spaces in between. And here's an example of the gridded system we used, showing the different sizes of grids. And on the right, example of the buffers, so each point representing each environmental data point. So for data on weather and climate, I use the NOAA's Climate Data Online tool to download daily weather observations across all coastal stations during the study period. So these were totals of daily precipitation as well as daily temperature averages. And all in all, there was over 400 stations. The NOAA Climate Data Online tool also offered 30-year climate normals, which have 30-year averages that describe climate. And during this process, we realized that there are significant gaps in some of the temporal and spatial coverage of these weather stations. So we also looked into this interpolated and gridded weather data set called DAYMET, which offers 1 by 1 kilometer pixels of daily weather observations for the entire US. We're interested in the effects of drought, so we downloaded some monthly drought indices. We're also curious about the potential effects of different climate teleconnections, such as El Nino or the North Atlantic Oscillation. So there we downloaded monthly values for those as well. To process the weather data, calculated multi-day running averages and totals of temperature and precipitation. That way we could figure out not only the 1-day precipitation total, but 5-day, 7-day, all the way up to 90 days. Same thing with temperature, where we could calculate those averages. These data are also summarized by season and year and compared with the 30-year climate normals. Here's an example of some of the outputs from that, where on the x-axis we have precipitation difference from normal, and on the y-axis we have temperature difference from normal. So here we can group different years based on their climate, with the upper right quadrant representing warm and wet years and the bottom left cool and dry years. And as right away, you can see that some of the most recent years of data were also the warmest. So those were data describing the physical landscape, weather and climate. We also needed data to describe the environmental quality of South Carolina's coast. So here I use data from CCAP, or the South Carolina Estuarine and Coastal Assessment Program. This is a DNR run environmental monitoring program that began in 1999. They collect data on water quality, which includes bacteria, dissolved oxygen, salinity, sediment quality, so different pollutants within the sediments, as well as collect data on the biological condition at each site. So here, small trawl net is run at each site where the nectin, which are the fish and large invertebrates, are sampled, and also with the same benthic samples that are used to measure the sediment. We get a picture of the benthic invertebrate community. And here's a map of the CCAP stations I used for this study. So I used data between 1999 and 2018. So 20 years of data. All in all, it totaled 811 stations. These were randomly selected to represent the full diversity of habitat across South Carolina's coast. And we're split into two basic habitat types. There's open water, which are sites greater than 100 meters in channel size, and tidal creek, which are those smaller. I should also mention that CCAP is run during the summer, so all the sampling occurs during July and August. So most of the data processing and environmental data were completed by DNR staff. For example, the sediment contaminant data, these raw values are converted into ERMQs, which are the effects range median quotients. These are used in environmental toxicology to describe the biological impacts of different contaminants, which are standardized along a biologically meaningful scale. From the trawl and benthic data, calculated species richness, which is the number of unique species in each site, and total abundance, which is the total number of individuals captured at each site. The benthic invertebrate data are further summarized with these two indices. So there's the benthic index of biotic integrity, BIBI, as well as the MAMBI, which is the multivariate ASTES marine biotic index. So these indices are a way to describe the health of the benthic communities. And this is important for this kind of research because unlike nectin or fish, which can move in and out of sites, benthic organisms are sedentary, so they really offer a good snapshot of the biological condition at that location. So once all these different data sets were collected and processed, they were then integrated across space and time. So to do this, I joined each CCAP station to its respective spatial unit, such as which watershed it was in. It's also paired with a matching year of land cover data. Each CCAP station was then linked to the nearest weather station that had available data. These were all combined into one relational access database for easy querying, filtering, and analysis. So for the data analysis phase, there's a lot to cover here, so we split it into two different stages. First, the exploratory data phase, which aims to answer the questions of how these data are changing over space and time. And then from there, moved on to the linear modeling phase. So here we can get at what specific variables are responsible for these changes, and also use these models to help predict the estuarine environmental response to climate change and increased development. So first, I was curious if any of the land cover data we had, what sort of information we could gather from that. So here is the change in land cover by coastal county, with the x-axis representing each coastal South Carolina county, and the different bars representing the amount of hectares of that county that changed or didn't change during the study period. And as you can see, most of the area remained unchanged. You can also see counties like Horry, where Myrtle Beach is located, having a higher proportion of land cover change. So within the area that did change, could also look at the net gains and losses of each land cover category. So what I have here on the right are these net gains and losses separated by each land cover category. You can see that measures of development, both high and low intensity, experience net increases during the study period, whereas measures of more natural land cover types, forest, agriculture, marsh, decreased. So what was interesting here is we did see an increase in the level of water. This could be the result of sea level rise or coastal erosion. So I was also interested in how these weather data might be changing over time and how well our data stack up to the predictions of the larger climate science report. So I averaged the annual temperature across the different coastal South Carolina stations and ran this generalized least square time series regression analysis, which gave us a significant positive slope, meaning that annual temperature has increased in the study period and seems to continue to increase. Also calculated the total annual precipitation. And while here we did see a positive slope, this slope was not significantly different than 0. I ran the same analysis on extreme temperature and precipitation data. So for this, I define extreme temperature as daily maximums in excess of the 95th percentile threshold, and precipitation as daily precipitation totals at that 95th percentile value. So I tallied the number of days that exceeded that per year and ran the analysis on that. What we see is that the change in extreme temperature is not significant, but days of extreme precipitation we're seeing an increase in those. So already, it's just this kind of crude analysis, we are already seeing the effects predicted in the climate science report. So I was also curious in how the environmental data might be changing over space and time. So here I use t-tests, which are a way to compare the means between two populations. For these data, I separated environmental data by the level of watershed development. This is defined by the amount of impervious cover each watershed, with developed watersheds exceeding 18%. So after splitting the data based on these categories, running the t-test, we saw that estuaries draining developed watersheds had higher levels of bacteria and sediment contaminants. However, measures of biological quality were not significantly different. So here you can see fecal coliform, metals, pHs, as well as all contaminants averaged across their ERMQ scores, showing higher concentrations in the developed versus undeveloped watersheds. So I ran t-test on after splitting the data up by time too. So here I split the data between early and late sample years, so from 1999 to 2008 and 2009 to 2018. And while measures of sediment contamination and water quality did not seem to differ between the two time periods, did see significant drops in measures of biodiversity between early and late years. So trawl species richness and abundance decreased in the later time period, and saw the same trend with the benthic species richness and abundance data. So I thought this was a pretty interesting trend and I wanted to take a deeper look. So I went back and ran that GLS regression time series analysis after summarizing the nectin abundance area and species richness by year. And what I found was a significant decline in both nectin abundance and species richness during the study period. Thought this was a pretty concerning trend, and I ran it on the benthic data as well. And once again, saw significant decline in benthic species richness over the study period. But interestingly, the abundance area remained relatively stable as it did not have a significant slope. What this could mean is that there's changes occurring within the benthic community structure. Perhaps there's specialist and pollution sensitive species that are being displaced by an increasingly disturbed habitat. And these roles are then being filled by more generalists and stress tolerant species. I do want to take a deeper look into some of the spatial trends in the data. I used hotspot analysis in GIS, where I summarized each environmental variable by its 14-digit HUC watershed and ran the optimized hotspot analysis tool. So this tells you how significantly higher or lower each watershed is from its surrounding neighborhood, with hot spots being significantly higher, cold spots being significantly lower. So these are PAHs, these are polycyclic aromatic hydrocarbons. You can see a hot spot right in the Charleston area and a cold spot down south. Ran hotspot analyses on some of the biological data as well. I thought this was pretty interesting with the benthic species richness data. So I observed this latitudinal pattern. As the further south you go, also observing higher levels of species richness. I believe this is because where south you go, you have higher tidal ranges, resulting in a more expansive and diverse estuarine habitat. So now we've shown how these data have changed over space and time, and moved on to the linear modeling phase, which could help explain what variables are responsible for these changes, and also build models that could be used to predict potential for environmental response to climate change and increased development. So as you can imagine, during this data synthesis project, created a pretty large and sometimes unwieldy data set. To handle these large number of variables, I use stepwise linear regression. This is an automated model selection tool that can create multiple linear regression formulas. It does this by adding and subtracting different input parameters and selects the combination that produces the best model. Here I define it as model with the lowest BIC. And all in all, there were 19 response variables, over 100 explanatory variables, all analyzed at 11 different spatial scales. So this totaled over 200 models, the final result. And here's an example of a multiple linear regression formula, where Y is the response variable, X representing each explanatory variables alongside an intercept, as well as a coefficient estimate for each explanatory variable and an error term. So out of these 200 something models, I didn't want to rely too much on this automated process, so I selected the best set of models to explore in greater detail and further refine. To this, I analyzed the residuals, removed outliers when appropriate, experimented with different data transformations, as well as explored interaction effects among the terms. In the final result, I selected 13 models that summarize water quality, sediment quality, and the biological condition. And here are some of those results. I selected 3 of these models to share with you. This is the model for fecal coliform. So on the left here is just an example of the formula, the response variable on top, and each explanatory variable, the plus or minus indicating the coefficient estimate, and the asterisk indicating the significance of those variables. So the fecal coliform, these were positively correlated with 2-day rainfall totals. This was interesting because we looked at several different time intervals for rainfall, and both the fecal coliform and the other bacteria model seemed to favor this 2-day rainfall mark. It was also positively correlated with impervious cover, so areas with more urbanization resulted in higher levels of bacteria contamination. But interestingly, it was also positively correlated with forest type. Fecal coliform, while most associated with human sources, can also have a wildlife source. It was negatively correlated with channel width and salinity, so more fresh and smaller channels at higher concentrations of bacteria is negatively correlated with marsh. This was something I saw in a lot of models, and I think it suggests that the potential for marsh vegetation serve as this kind of buffering effect. And it was also negatively correlated with one of our drought indices, PDSI. So what that would mean is that in periods of drought, high rainfall events are responsible for these bacteria entering the waterways. And on the right, these are added variable plots. So this is a way to visualize the relative effect of each parameter when controlling for all the others. And the point of these graphs is just to show you the general shape of the data, as well as the significance of the trends. So the top left, you have salinity term, having a pretty steep line, suggesting that salinity is a pretty important driver of the variation within the model. Whereas this PDSI, which is the drought, it's a relatively flat slope, meaning it's not as important of a predictor, while still significant. And then model statistics, the adjusted R squared describes how much of the variation the model explains. Here we had a value of 0.46, meaning 40% of the variation within the data can be explained by this model. So these were data on the ERMQ scores averaged across all the contaminants we looked at. It was positively correlated with the silt and clay content. This is something we expected, as clay particles have a tendency to attract and hold on to different contaminants. It's positively correlated with population density, meaning more populated watersheds at higher levels of sediment contamination. It was positively correlated with winter precipitation and negatively correlated with winter temperature, meaning with cool and wet winters resulted in higher levels of contamination. This is another interesting finding. So this was not the only model where winter precipitation occurred. It actually occurred in a lot of the models. We're trying to figure out really what could be behind this. One explanation I had is perhaps these contaminants are being washed in the estuaries year-round, but during most of the year, they're being intercepted by an active biological community and broken down. And in the winter, when temperatures are cool, these microbial processes might go dormant, allowing these contaminants to enter the estuaries and eventually settle onto the sediment. These are also negatively associated with group A soil types. So these are well-drained soils, meaning that watersheds that are well-drained are less likely to have the runoff that could contribute these contaminants to the estuaries. And here our adjusted R square value is 0.74. And our last of these models was MAMBI. So this is one of the marine benthic indexes we use, with higher MAMBI values representing more intact and healthy benthic communities. These were positively correlated with channel width and salinity. This is also to be expected, as more open, saltier channels are typically going to be the more marine environments where this index has been calibrated for. They're also positively correlated with one of the development categories. So it's developed open spaces, so think lawns, golf courses, and fields. So I'm not really sure what could be responsible for this, but kept in the model because it was significant. Positively correlated with 45-day precipitation totals, negatively with the silt clay content, negatively correlated with development of high intensity, so the more urban landscape, also negatively correlated with the 90-day temperature average. So both warmer and drier conditions might result in lower benthic health. And here we had an adjusted R squared of 0.47. So some summary from these models, what we found was that physical habitat, or that salinity, channel size, or sediment composition, is responsible for a large proportion of variation within the response variables. Measures of development were correlated with higher levels of bacteria and higher concentrations of sediment contaminants. Precipitation was positively correlated with higher levels of bacteria and sediment contamination. This supports our runoff hypothesis, but the effects of temperature were less clear. Biological responses were more dependent on weather and climate variables than they were with the land cover, and warmer and drier conditions were often correlated to lower biological quality. So before moving on to the predictive modeling, just wanted to take a moment and share some of the meta-analysis we did. So as I mentioned, with the stepwise process, created over 200 models. I think there's some really good information just in what parameters showed up in those and patterns that were revealed. So I calculated the frequency of each explanatory variable that occurred during these models and found that variables of physical habitat were the most prevalent predictors. I think this is important because we're trying to explain a very diverse landscape. I think it's really important to include things like channel size and salinity in the models. Impervious cover was the most frequent land cover variable that popped up in these models, followed by marsh. And out of all the weather and climate variables, once again, this winter precipitation, winter temperature was the most frequent. And also curious of sort of model performance. So we used the R-squared values. They were summarized by model types, as well as the spatial groupings. And what we found is the 2-kilometer buffers were best at describing models of water quality, so things like bacteria. This suggests the effects of land cover are much more local for these. And sediment quality, which were most influenced by 10-digit Huck watersheds, suggests these are occurring at larger spatial scales. And biological quality is overall a lot more varied, but 12-digit Hucks seem to do the best at describing those. So those are the smaller subwatersheds. So the next phase of this project was predictive modeling. We built all these multiple linear regression formulas. We know that the landscape is changing, the climate's changing. So how can we use these models to make predictions of the estuarine environment? So to do this, I researched different projections of population growth for coastal South Carolina, as well as consulted with the Fourth National Climate Assessment Report to get different projections of temperature change and precipitation change. These were aimed at changes by the mid-21st century, which is the year 2065. And with these data, I created 14 scenarios of different combinations, went back into the data set and adjusted the different explanatory variables according to these different scenarios, and then reran those multiple linear regression formulas to come up with these predictions. And here are the results of those. So this is that fecal coliform model again. On the x-axis, we have the different scenarios of change. So high and low population, high and low temperature increases, as well as precipitation increases, followed by different combinations of these changes. This horizontal line represents baseline conditions, with the dotted lines indicating the 95th confidence interval of those predictions. Each point represents the predictive model outputs, and the error bars also represent those 95th confidence intervals. So in instances where these error bars are overlapping with the baseline, I think it's a little harder to say what the predicted outcome could be. In this case, we saw significantly higher predictions of fecal coliform given population growth, but the effects of climate change were more subtle. Compare that to the Enterococcus model. So this is another indicator of bacteria used to measure water quality. And here we saw the reverse effect where increase in population did not significantly change the predictions, but changes to temperature did. So fecal coliform might be more sensitive to changes in land cover, whereas Enterococcus bacteria could be more sensitive to climate change. And here, while precipitation was included in the models, their increases did not significantly change the outcome. Here are the predictive model results for the ERMQ score, summarized across all contaminants. So we saw that population growth increased the predictions of sediment contamination, whereas increased temperature was linked to actual lower predictions of sediment contamination, and precipitation resulted in less than significant increases. For the biological data, this is showing the predictions in nectin abundance area. So here, population changes resulted in slight declines in abundance, and increases in temperature result in pretty significant predictions of abundance. And precipitation, this model didn't have a precipitation term, so they are the same as baseline. For the benthic community, these are estimates of the benthic species richness. Under the population growth scenarios, we're actually saying increased species richness. I think this could be due to the fact that those measures of low intensity development were positively correlated with some of these benthic models. Increases in temperature resulted in lower species richness of the benthic, and precipitation increases resulted in slightly higher, but not above the baseline condition. So this predictive modeling revealed important drivers of environmental change, as well as the sensitivities of different explanatory variables. Increased development could result in higher concentrations of fecal coliform bacteria, higher levels of sediment contamination, declines in nectin abundance, and increased benthic species richness. Temperature increases might be linked to higher concentrations of Enterococcus bacteria, as well as declines in the nectin biodiversity. Now, precipitation increases, while precipitation was a significant predictor in many of these models, in this predictive modeling practice, they were not as influential as expected. I think this also revealed the potential for compounded effects. So while increase in precipitation might not be significant on their own, when combined with things like increased temperature or development, they could have some impact. So going back to our original hypotheses, how well is our data done to explain these? So the first hypothesis, which describes the effects of land use on environmental quality in estuaries, we saw that measures of development are correlated with higher bacteria concentrations and higher concentrations of sediment contamination. However, the effects on the biological community were more varied and more difficult to define. Our second hypothesis, relating to changes in weather, what we saw is that precipitation was correlated with higher levels of bacteria and sediment contamination. This could very well be pointing towards runoff as the source of these contaminants. And temperature was often negatively correlated with measures of biodiversity. Perhaps this could be one explanation for those significant declines that we saw in the time series analysis. And overall, I think this project has revealed the importance of long-term environmental data sets and demonstrated the effectiveness of a data synthesis methodology. And while South Carolina is still a largely natural landscape, the effects of development were still observable and felt at large spatial scales. What I found was South Carolina's estuarine habitat is incredibly diverse and was not easy to summarize with a one-size-fits-all model. Lastly, the estuarine environment has been influenced by land cover and climate. This type of modeling can help understand these relationships and demonstrate the potential consequences of climate change and continued development. So this was the last phase of our flow chart that I showed at the beginning of it. So what do all these findings mean for policy and management for coastal South Carolina? Well, this region is going to continue to grow. I think it's shown, provide more evidence for the importance of ecologically focused stormwater management as being central to sustainable development. These include best management practices and low impact development designs, such as green roofs, constructed wetlands, permeable pavement, and rainwater harvesting. While climate change may be more difficult to address as it will require global cooperation in reducing carbon emissions, this type of green infrastructure, much of which can be done in your own backyard, can at least help create a more resilient coast ready to face these challenges. I wanted to acknowledge funding sources. This project was funded through the South Carolina Sea Grant. All the support I got to attend different conferences, received funding from MESA, the graduate school, as well as DNR. A big shout out to my committee members, been super supportive, all my coworkers in the environmental research section, DNR, my peers and professors, the environmental studies program, of course, my friends and family. Here is the contact info. If you have any more questions or thoughts or ideas, as well as my GitHub account where I have all the data stored, as well as the different R code I used for the analysis and to create the graphs. So I'll open it up to questions.
Thank you, Lloyd. Great job. Yep, like you said, we'll open up to questions. So if anybody has a question, please go ahead and type your question into the chat dialogue. Or if you prefer to ask your question verbally, please just type that you have a question so I can call on you. No questions? There's got to be some.
Well, I was waiting for someone from the general audience to maybe ask one, but as a committee member, maybe I'll jump in. Really great job, Lloyd. That was so interesting. And I think you brought together so many sources of information and compiled it in such a way that you've created a very valuable data set for many stakeholders. And I'm sure you'll leave a legacy where people will be continuing to learn from your work into the future. So that's really great. One of my questions is, I was wondering about your predictive model.
Mm-hmm.
I was wondering, what is this, how did you derive the 95% confidence intervals? I'm a little bit shady on that. And what does it mean? Like, what's the source of the uncertainty in those predictive models?
Right, so that goes back to the those models that I shared with multiple regression formulas. So I went back to the data set that I used to create those models, and then used those different change scenarios and adjusted the different explanatory variables when appropriate, and just reran the model. So I used this predict linear model function in R, and that confidence intervals are derived from those error terms that were included in the multiple regression formula.
Okay. And just as a quick follow-up, I don't know, if you were to explain, you know, the results, the major takeaways to various stakeholders about this predictive modeling, as I'm sure people who are managing this growth might be interested in, you know, how would you represent the uncertainty to them? And would you want to add any other sources of uncertainty to your conclusions?
Right, that's a good question, because I think that's something difficult for science communication, showing how there's always level uncertainty. So I think it's important to pair those predictive models with the in-depth linear modeling that I did earlier. So I think you wouldn't want to show them alone, but pair them up with all the work that went into creating those original formulas. And there you can show sort of the R² value of how much variation we'll explain with these different terms, and also give some sense of the rigor that went behind the predictions. And I think also another take home of the predictive modeling are just the different sensitivities of each predictor. So yeah, precipitation is positively correlated with many of these variables, but when we tweak them with the predictive modeling, it didn't seem to cause a significant increase. That still doesn't change the fact that these are still positively correlated, and this is just a very generalized approach. Perhaps if you take a deeper look into it, you could start to find, where all that variation could have some impact.
Thanks. Looks like Sharleen has a follow-up.
Yep, I was also curious about that connection. Oh, first, let me say, Lloyd, great job on the presentation, great job on the thesis. You did a lot of wonderful work here. So I was curious, because the linear regression models that you created had some very subtle expressions or formats of precipitation data and also population data. So it wasn't just, a certain amount of precipitation that occurred in a year. It was, for fecal coliform, it was the 2-day rainfall, it was a drought index. And then for population, I was curious how you translated a % increase in population in the way that it was expressed in your linear regression models, where it was more like impervious cover or % of certain land cover types. So if you could explain how you went from those the linear regression models to the predictive models when they kind of had very different types of parameters.
Right, that was definitely a challenge. So with the measures of land cover, I simplified it and just made the assumption that increases in impervious cover and measures of development would be a one-to-one relationship with population growth. And while I didn't really have any hard data to back that up, during the exploratory data analysis, I did see those sort of parallel lines of growth. So I could have kind of created a more elaborate formula to maybe figure out the exact relationship between population growth and levels of impervious cover. But for this purpose, I thought it was good enough to just assume this one-to-one. So if it was a 130% increase population, you could also expect a 130% increase in impervious cover.
Okay, so that makes sense for population. And definitely when you're doing modeling, you need to simplify things in order to make it work in a model. How did you handle the rainfall? Since the time period, like sometimes it was seasonal rainfall that was the most important, like winter rainfall, for instance, or 2 days rainfall before the sampling date. How did you incorporate that into the predictive model?
Okay. Yeah, so for the sort of the observed rainfall totals, which were those like 2-day up to 90-day, those were just increased by %. So if it was a 10% increase in rainfall in the projection, I just increased all those values by 10%. But for the seasonal, I went back and calculated sort of the seasonal averages, which these were defined as, in the modeling, as difference from normal. So then I kind of back calculated what that adjustment would be, and I actually added X number of centimeters of rainfall or X number of degrees Celsius to those deviations from normal.
Okay, great, thanks.
Looks like we have one from Abby here. Would you be able to break up the data or models by municipality so that the local government staff or officials who might be less familiar with the HUCs could easily see results for their area?
Yeah, I think it's a really good point. So we really created these models that were pretty general that could be applied to the whole coast of South Carolina. And I think we made some sacrifices there where if we were looking at more localized things, we might end up with better models. But I think in general it's important to kind of create this generalized approach. But I could easily, in GIS or whatever, just subset it down to a specific county and run a lot of these same models and probably get different results, but then it'd be harder to apply that elsewhere. So I think kind of one of the motivations of this project was to keep it general on purpose so we could apply it to the whole coast.
Any more questions from the general audience before we move on to the committee meeting?
I'll just wish a big congratulations to you, Lloyd, and thanks to the committee for all the work that you've done on Lloyd's project.
Appreciate it.
All right, well, thanks again, Lloyd. And we will head into the committee meeting. So if you're not on the committee, if you could please excuse yourself. Thank you for attending.
I think it's my committee plus Dad. Okay.
Man, I'm on mute again. Andrew, you are now the host.
All righty, thank you.
Okay, bye.
Bye.
Good job, Lloyd.
Okay, good job, Lloyd. Here we are with distilled down to the committee. Tim, I know you have a time commitment coming up, so if you have any questions, the floor is yours.
Sure, and I'll be brief. And also I'm dragging Barbara with me for this other. Barbara, you can be late. You've got a good reason to be late if this keeps going. Anyway, I echo the sentiments already provided, Lloyd. Nice job. This is a tricky task. I'm sure you were thinking many times late in the night trying to pull all this together. And I completely agree this will serve a lot of people in the future. This is the kind of collective resource off the shelf kind of thing that a lot of people really like to have. And it's a good point there, Abby. I liked her question about converting some things into site-specific off-the-shelf info. That would be really helpful. And I guess my first thing is, along those lines, do you see a product, kind of a spin-off product, possibly, in the spirit of what Abby was asking? Is there, like Sea Grant likes to do, make brochures and pamphlets and things like that. I'm not saying that's part of your job or anything, but what would be maybe the standout aspect that you could have into a two-page flyer or some kind of brochure for non-scientists? Would it be what you concluded on, the green infrastructure aspect? What can people do at the local level? Or is this an avenue that you've opened for people to think a little more deeply about data and analytics, or maybe both? Just want to get your perspective on that.
Right. So, yeah, I think all the stormwater management solutions, stuff that I ended on, it's pretty approachable, but that's kind of wasn't really the focus of this project. That was just kind of an afterthought. So yeah, I think the biggest takeaways is really just this kind of approach really hasn't been pretty new territory here with this data synthesis. So we're just surrounded by all these different data sets. These weren't available to researchers 10 years ago. So I think it's a lot of uncharted territory. I would like to, I think, focus on that aspect, just sort of the potential for data synthesis, like large, I wouldn't go as far to say it's data mining, but just sort of the products that are available that anyone can do from their, computer if they have internet.
Okay.
But yeah, as far as like a short brochure, I think driving into these specific weather and land use variables. So one of the take homes that I mentioned in my discussion, but didn't really come up here, is the presence of marshes being this sort of buffering effect. So saying these ability for these natural land cover types to help possibly filter out contaminants.
Mhm.
Also just the level of development, how it's even in the more rural parts of the coast has shown these different impacts. And also this weather component of compounded effects of precipitation, whether it's be like extreme precipitation events on top of a landscape that's becoming increasingly urbanized, and the potential for runoff becoming a more serious issue.
Well, can I, just as we're talking about this, and maybe you've done this already, a story map or a series of story maps. Again, not putting this on your shoulders to do this, but maybe aspect that could get into the future work part of your thesis or something, or just food for thought for Andrew and the group. Maybe building a couple of different types of story maps or providing information and guidance to people who want to do that kind of work could be pretty useful in an ArcGIS type of or an open GIS framework, where people then can access the information in a visual way. Some of the products, the figures you put in your thesis are really valuable. And to have that into more of a dynamic ArcGIS space might be pretty helpful for some of those folks.
Yeah, no, I think this would be a perfect use of a story map. I didn't cross my mind until you brought it up, but that's something I worked on in one of my GIS classes. Also like our Shiny app, that was something we went over pretty briefly with Dan McGlynn's class. So there you kind of have all your input parameters, sort of dial up precipitation, see what the effects there would be, change levels imperviousness, salinity. That's all doable with the data that we already have. Formatting into application would be a lot of work, but I think I set the foundation for that.
Definitely. And then just one last kind of detail about the data and statistical methodology, I guess you could call it. So that 4-panel graph where you show warm and wet and warm and dry and all those cross-pollinating the temperature and the rainfall. I was kind of going back and forth making comments in your thesis document, and I'll share that later. And again, I'm not beholden to going one way or the other, but what struck me is the fact, for example, 2015 heavily weighted on the 1,000-year storm in early October. And so otherwise that year may have been deeply embedded in the warm and dry panel or of that graph. And then only because of that large rainstorm event is it over on the upper right-hand panel, the wet side of it. So, you know, extreme events are important, and you can't pull that out in the sense of, obviously it happened. And so that then moved a lot of sediment and fecal coliform and Lord knows what else through the runoff of that. And so it's important, and it informs upon your whole situation. But if there's any sort of qualifier, you could, I don't know how to present this. Again, I'm thinking out loud that how to show and emphasize in those kind of diagrams or figures the impacts of extreme rainstorm events. You know, so your annual result is that particular year ended up warm and wet, but all but 2 weeks or 3 weeks of the entire year were pretty dry. So anyway, just wanted to put that out there, and I don't have an answer necessarily, but if there's any sort of annotation or footnote to put on graphs like that, and you can make a case for 2017, 2016 as well, where only annual rainfall totals were heavily weighted by hurricanes. So just wanted to throw that out there.
Yeah, I agree. It's kind of hard to capture those more extreme events. You know, a lot of these weather observations kind of got lost in the wash, you know, during this process. But I think that's something that I mentioned to Andrew wanting to explore a little bit further was
But you
a
did,
way to include intensity. in the.
Did do that by that same graph by season as well, isn't that right?
That helps a little bit
Make
on.
it a little bit more granular. It's still not perfect. And then I suppose, you know, again, I'm not trying to add additional stuff for you to do, Lloyd, but you have the extreme event data. You know, you could do a similar graph looking at a number of extreme events per year or something like that too, as another way to get it. Yeah, I agree. There might have been a lot of rainfall total that year, but
Yeah.
it's very patchy.
Well, in this particular situation's on my mind because we did a study of Francis Marion National Forest, one of the watersheds, Turkey Creek, that drains off into the east branch of the Cooper. And that 2015 storm event from Hurricane Joaquin resulted in 78% of the nitrogen TMDL. The load of nitrogen for the entire year was, you know, more than 3/4 of it came from due to that one extreme event. And, you know, again, that's not necessarily captured unless you get down to the event-based results. And again, that you were doing a holistic analysis on this, so I'm not trying to be a Monday morning quarterback or anything, but something to the effect that you could, for future researchers to investigate those kind of large storm event impacts, I think could be pretty interesting. And I think most of the analytics and techniques you used would be applicable down to that storm by storm basis, I think. But just something for someone to consider going forward.
Yeah, for sure.
And that was, you know, I've got some other notes in the Word doc, and I'll share that back to you in the next few days. But
Yeah, looking forward to it.
nice job on this.
Thanks.
All right. Okay, I'd add a couple questions. Tim, are you good?
Yeah. Oh, yeah.
Okay, cool. Well, thanks for coming.
Yeah, I'll hang out as long as I can and may bail out here in a few minutes.
I had a question, Lloyd. So you ran these prediction scenarios on kind of like the average condition, is that right? You know, were you able to get any insight into, you know, we've talked about doing a map of these predictions or something, into how, say, a tidal creek might respond differently from an open water, or a more saline site might respond differently from a more brackish site? Have you gained any insight from that? I mean, we had all the pieces are there to do that. You know, do you have any thoughts about that?
Yeah, and that was something included in the discussion, is this idea of sentinel tidal creeks, where they're kind of the first ecosystems to show impacts from development. And yeah, there were some early on in the exploratory data analysis phase. I used this t-test to kind of compare the different environmental variables between the tidal creek and open water sites, and generally did see that tidal creeks were more polluted and degraded than the open water counterparts. And so I ran all the predictive models I shared during the presentation. Those are summarized across all habitats, but in the thesis itself, I did split them up into data tables to see the potential differences. And what I found there was that when I averaged the percent change, so I compared the prediction to baseline, got a percent change, and averaged it between both the tidal creek and open waters sites to see if maybe one was more susceptible than the other. And sort of saw the opposite effect, where the open water sites did seem to change more from the predictive modeling than tidal creek sites. So, you know, I did have some evidence that maybe these smaller headwaters are more susceptible to change, but during the predictive modeling, it seemed to provide evidence to the contrary. So not really sure, don't have any solid answers there, but I think once again, breaking it down into smaller habitat types might result in different models. Maybe the open water and tidal creek, maybe we need to go even have finer resolution, break it down into freshwater tidal creeks versus these saltwater or more intermittent ones. But yeah, I think there it's kind of the same thing with Abby's comment about the more specific you get, the less you can start to apply it across the board. But definitely food for thought there.
Cool, thanks. Another question I had, so I mean, you did all this modeling, we found just about every variable we could find. I mean, we didn't see some relationships that maybe we thought we would see. I mean, are there other variables that, you know, if we could go back in time, you would like to, you wish we could have added or something like that? Or do you think there's anything we're really missing here?
Yeah, I think it's a little lacking in the water quality. So right, at some point we had to say these were enough variables to, you know, kind of had our plate full already. But I think definitely taking a deeper look into the potential of nutrients and the effects of eutrophication, which is a pretty serious side effect of stormwater runoff. Kind of drop those early on because there's just kind of a lot of noise in that data, but taking a deeper look into that and trying to get the sort of what nutrients are popping up and if these correlate to, you know, higher levels of the chlorophyll, more hypoxic events. And also, I think the biological data definitely deserves a deeper look. So I just kind of lumped all nectin together. When I looked at the most frequent species that showed up, they're mostly shrimp in the abundance counts. So what if I controlled for that, removed shrimp, or just looked at shrimp alone, just fishes? I think we could get a more complete story of what's happening in the biological community after doing that.
Mm-hmm. Cool,
But
thanks.
yeah.
Any other questions from the committee?
Yeah, I had one. It sort of struck me that the biological responses should really be also dependent on the water quality and sediment quality. Sort of in your models, you didn't have those as part of independent variables in your biological model, I think.
Right.
And I was wondering what you thought about the ability or the value of adding those in, or why you decided not to include those.
Yeah, and I started to do that with some of the benthic data, but you know, I kind of through this process wanted to keep the response variables separate from explanatory variables. And once I started including, you know, there is some gray area where it's like, yeah, these are both explaining the same thing. Still wanted to keep them separate just for continuity throughout the project. But definitely, I think I was trying to get at essentially what's driving the changes. And if we can get to, you know, specific measures of development or specific weather phenomena, I think that gives a better message than perhaps like lower DO, because ultimately we would want to tie that back to what's happening in the upland watershed. So I wanted to kind of keep the focus in the uplands where there's like effects to the land, and there's also effects of weather and climate.
Mm-hmm.
I agree. I think they're definitely related, and that was one of the limitations of this style of linear models, is you can only pick one response variable. There's a whole nother world of statistics out there where you can start to combine these different things and start looking at it multivariately.
Yeah, and just to add on to that, another example like that might be the ERMQ. I mean, if that was a response variable for most of the time, but maybe that's driving toxicity factors for the organisms or something like that. So
Exactly what I was thinking.
yeah, interesting.
And you know, I like your response there, Lloyd. Like, you know, you're looking for the drivers. Like, what is the change that's catalyzing? Maybe the change in water and sediment quality. I see that. But maybe you could just mention in your thesis, like, I think at one point you say the biological model was less predictive than the others.
Mm-hmm.
And maybe that's why, because you have this other noise of uncertainty, even, you know, how those end members of temperature and precipitation are driving changes in water and sediment quality, which then go on to affect biological quality. So the fact that it's maybe an indirect causal factor, you have like two layers of causal factors for the biological model, means that they're, quote, less predictive, or you had harder time describing all the variation?
Yeah, no, I think that was one of the more frustrating aspects, was trying to, seeing this decline. It was pretty concerning in the biological data. But then when it came down to trying to explain it, started to lose some of that.
The power.
Right, but
yeah, because the declines were a lot more obvious for the biological communities than for this for some of your environmental quality data.
yeah, so I think there's something bigger at work.
Yeah, I guess I was also wondering, since this is a technical thing, the Welch's t-test assumes normality, I think, correct?
Yeah, I think so.
Distributions. So do you think some of your results are affected by this assumption, or what made you decide to use those types of stats rather than one that doesn't make that assumption?
Well, the Welch's t-test versus the student's t-test at least has the benefit of being able to compare populations of different variants, so different sizes.
Mm-hmm.
So here, it wasn't a symmetrical distribution where half the data were in this group and half the data were in the other. So Welch's t-test allowed for that, like loosen that assumption. But yeah, I think this was something we ran into the linear modeling too, is, some of these data just weren't perfectly behaved. And I think at some level we just had to relax the assumption that these residuals will be perfectly fit a normal distribution, or I think with the t-test, but
well, you're talking about residual variances, right? Which is one thing, but then like your distribution of observations would be normal or not normal, and you chose to go with stats that assume normality.
right.
Maybe because some of your data sets are normal, but I was just noticing that maybe some are highly skewed, and maybe your significance is affected by that fact. Like for instance, with I know it's like DDT was one of them that looked like it was highly skewed. And so maybe if you chose a non-parametric stat, again, I don't know how if it would change the outcome, but maybe you would see some differences in the outcomes there.
Right.
I don't know if it's worth going back in terms of your timeline and checking this out for some of the data sets that look like they might be skewed. If you just look through your tables and then say, okay, well, let's just try a different type of test instead. Use like Wilcoxon sum or something else that doesn't make that assumption. Then you might change some conclusions. Like, and the DDT, for an example, like it was different, I think, between early and late times, but it was small. It was like a p-value between 0.1 and 0.05.
Mm-hmm.
So you might be reporting on something that actually is like a type 1 error because of the assumption of the stats and the p-value.
Yeah, no, it's something worth taking another look at. I mean, I did experiment with different transformations of the data.
Right.
Tests.
Oh, you mentioned that. So did that help anything?
It did here and there. I don't think it was consistent.
Right.
But yeah, in some cases, log transforming, especially the bacteria data, really helped with the distribution and made it a little more normal. But
so you did log transform some things and then do that?
Mm-hmm.
Oh, well, I missed that. Did you write about that? Did you write about that in the methods?
Somewhere. Well, the axes, when it is log transformed, it is labeled in the different figures and tables. I don't know if it's...
And you met the assumptions of normality in some cases and had to log transform.
I don't think I went through that rigor with the t-test, but definitely when I was creating linear models, I experimented between untransformed versus transformed and try to make a decision about sort of the distribution of the data and residuals there. But with the t-test, I don't think I was that rigorous with yeah.
Okay. I guess that's one of my major recommendations then, is just to go back maybe and check some of that assumption.
Right.
We did do quite a bit with exploring transformations in normality on the linear modeling part. And we, with so many different data points and so much biological data, especially, there was a lot of concern about non-normality. And so highly not normal things we basically picked the better models, the models that we felt were more closer to that, if not completely normal. And then we met with Alan Strand in the marine biology program, and he gave us a little bit more confidence in interpreting those, in that as your sample size increases, there's increasing evidence to suggest that the true, like, proper, Shapiro-Wilk normality or whatever is less concerning than looking at the data and being confident that it's good data and that, stuff like that. So
yeah, that's true. Like, larger data size, your sample approaches the population.
right.
maybe if you see yourself going down a hole, maybe just check a couple and see if it changes some of those major conclusions about significance. Maybe that's where you need to focus.
Right.
But you could always just write that. If you didn't, maybe I missed it. The methods, like, we use the law of whatever, large sample size and assume normality.
Sediment stuff, the ERMQ stuff was the best for sure. Like, some of those were just really good, like straight and transformed.
Yeah.
And I think as we got into more of the biological stuff, it's where it became a little bit more like a little dicier, so that's why we had a harder time finding good models and good models to share on that.
Makes sense. Thanks. I do want to have, I know Sharleen hasn't talked a lot, but if I could just dominate for a minute before I have to go to my department meeting. I will have to say, I was really impressed with some of the linear models. I mean, all of them, I guess. How the result, I think, really matches up with what you would think the mechanism could be. Like, you didn't have any weird signs where you're like, that doesn't make sense. Why is that indirect? Like, a lot of them, like, you can basically say, yeah, this makes intuitive mechanistic sense on why this is behaving in this way. So it was really awesome to see. I was especially interested in some of your, like, organics, ERQM and et cetera, causal factors. So I might type this up a little bit more in an email, but like, your ideas about the winter precipitation and temperature are pretty interesting. And one thing I might add to what you've already written, possibly, is that with more precipitation, you can have more wet deposition. And I don't know to what extent wet deposition is important. I know it's important for pHs, but I don't know about DDT and PCBs in this region. But that could then sort of work its way down to sediments, I guess.
Does wet deposition, like, transfer from the atmosphere? Like,
yeah.
Just because it's wet outside, it's like stickier in a sense, or?
Yeah, so the other thing that matches, and I wouldn't normally bring this up, this is usually seen in soils, but sediments, I don't see literature about this, but when it's warmer in the winter, you'd expect less PAH emissions because there's lower heating, and heating of buildings is one of the primary emission factors that can describe seasonal changes. So you have warmer winters where you see less pHs in sediments, right? And you also see wetter winters, and that's where you'd expect more of capture from the atmosphere, and then bringing it down. And usually, like I said, usually you see this for soils, but maybe it's happening for sediments too.
That's interesting. Thank you.
And then I also agreed with your plants and biological mechanism to an extent. Unfortunately, because PCBs and PAHs and DDT are less biotransformed, right? And so if you were looking at nutrients, then this idea of lower activity and less transformation would make sense. But maybe instead what it is it's a die-off, where these plants and you know, other matter is storing organic contaminants and then dying off and releasing it. And so I think I have another paper about that that I can share with you if you wanted to add that to your explanation. But
Yeah, no, that's all really interesting because I was kind of just making well, guesses at some point, but
well, no, but what I was saying in the beginning was like, these are, you know, really good, and your descriptions are good in terms of the, you know, the disturbance remobilization hypotheses is really good, I think. But you know, just ideas for adding to it those weird, I guess, observations that you weren't fully sure how to explain, I think have other explanations.
That's interesting. Thank you. One other, what you said reminded me of this discussion that we had in the past. You know, we have this suite of contaminants that we've analyzed in the sediments. Some are legacy compounds, you know, and some are still actively, you know, emitted or deposited. I mean, as far as mechanisms of transport to the estuary, I guess the plant seasonal die-off hypothesis could apply to either of those, you know. I don't really have a specific question, but that was like two things we were thinking. We have some variables that are like, you know, these like a banned pesticide or something was applied a long time ago, and how do we explain the mechanism of transport of that versus something like PAHs or I can't remember what specifically they were right now, but you know, things that are actively still being deposited to present day. So
yeah, and sort of I don't know what's been seen in sediments. It surprises me that you see some degree of change in the sediments, which is thought about as sort of as a long-term accumulation reservoir. So I think it's interesting that you see this change, because usually these sorts of observations are seen in soils and air, not sediments. But like you said, something, you know, maybe worth looking into more in the future.
Related to the PAHs, there's actually we have a whole bunch of different PAH parameters in the CCAP database based on the sediment data. And one of them we remove in one of the PAHs we remove because it's produced by, I think, natural fire cycles in pine forests or something. And so I'm not sure in this data set which PAH parameter we used. I think in the ERMQ, it doesn't necessarily exclude the naturally occurring PAH, which can be a very considerable contributor to that overall number. So yeah, that was just something that came up. And another kind of different topic, but something that Barbara was referring to also, is dissolved oxygen is very important for biological communities. But the dissolved oxygen we were using was just from a 25-hour period when we had a logger out at that site at one time point. So I'm not sure we could necessarily use that DO value as a strong predictor of what's going on year round or what those organisms are exposed to during their entire lives. But it'd be really interesting along those lines to actually collect samples longitudinally at some locations, like at the near sites where they're continuously monitoring water quality for a long period of time, or at the Socorro site that we recently established somewhere near there, and USGS water quality sondes, and then actually like do year after year with different climate parameters, or weather parameters rather, and see how those do change. Because one of the problems with CCAP, it's very useful to have this randomized selection over the whole coast, but it'd be really nice to be able to look at changes in a single location with weather and different factors, and watching maybe development happen in certain watersheds if we were lucky enough for that to happen, where we had continuous water quality data available. So yeah, that might be kind of a future work sort of thing as well.
Sharleen, you could write a grant.
Okay.
The money.
I will do my best to bring more money into ERS. Stepping on Lloyd's shoulders here.
Yeah.
Or standing on.
Coming in.
Yeah, that was just a, it's not exactly follow-up, but about the CCAP site selection, the randomization. I was wondering if you think that the CCAP site selection could have influenced some of your temporal trends in the contaminants, between late and early years. I noticed that there was a contamination peak around 2010.
Mm-hmm.
And I don't know, I was wondering about that.
I think that's possible. So even in the course of the data we have, there was a change in sampling methodology where they were sampling 40, 60 sites a year and down to 30 now.
Yeah, it was between 50 and 60 from 1999 to 2006, and then funding dropped and it cut in half down to 30 from 2007 to present.
So right, I think there may be some effects where more recent years we're not getting as good of a random sample, but I think this is just nature of this ecological monitoring is.
Oh yeah, no, I don't fault you. I noticed there was a peak there.
I think there's a possibility for just
Yeah, I mean,
where
so
the ice landed.
they're selected through a stratified random design to cover the whole coast. But yeah, I suppose that even with a random design, it's possible that there could have randomly been sites selected that maybe made that peak stand out.
Yeah, and I think maybe you take care of it when it comes to your linear models, is because you would have hopefully maybe accounted for some site characteristics that might have resulted in that peak, which I think is a nice outcome of the way you designed your work. But it might affect your t-tests, not your model. Well, I probably need to go and join my department meeting, but I don't have any other questions either.
Cool. Well, great. I really appreciate your input here and gave us some good insight. What's going on? Some of these contaminants. Your expertise there.
Do we need to have like a closed session?
Oh yeah, yeah. Yes, we do.
Sharleen, did you have any other questions?
No, we already spoke on Monday with a practice presentation, so I've kind of asked a bunch of things already.
I figured. I guess I could stick around for a brief closed session.
Okay, so I now have the power. Okay, Lucy gave me but Lloyd, I'm going to move you to the waiting room.
Okay, so.
Good job, Lloyd.
Oh, sorry, that was a little abrupt.
That's okay.
Okay, so he's in there. So we don't have Tim here. I don't have any further questions either. Appreciate your input, Barbara and Sharleen. So do we need to formally come to a consensus?
I feel pretty comfortable that he nailed it.
I do too.
So this is just the defense. And then is there a separate sign off for the thesis document after he makes edits? This is my first time through the process. I'm not quite sure with all the paperwork for MES.
Yeah, there are two forms.
Okay.
I think there's a thesis defense and the title page of the thesis.
Right. Okay.
So this is there's the thesis defense part, and then there's the approval of the thesis itself. So right now we're just saying whether he passes thesis defense, and then it's still contingent upon his completion of the edits and to satisfaction of the committee.
Yep.
Correct. Okay.
And then what is the deadline for that? Like, how much time does he have to make edits after today? Because there were some I have a bunch of small edits, and then there were some larger suggestions as well that could take longer. And so just deciding kind of where in the range to do that cutoff of, yes, this is the final thesis document versus what publication might come out later.
I don't know what the deadline is. I'm sure Lloyd knows that if no one else does.
Lucy about that. So.
Yeah. But reading through this, I thought it was so well organized and written. I really was impressed. I don't know how many drafts he went through with you, but it's quite good.
This was the second draft, but we've been working from an outline for a while, so we were making sure that it was kind of organized, and at least organized in the way we were thinking about it. So I'm glad to hear that from you, that it seems logical. And
yeah, it does. The only I saw Lloyd in the hallway, and we joked around a little bit, socially distant with our masks on. But he said this summer was like a horrible writing retreat. Because he was isolated and just writing. Well, I guess that's one positive outcome.
Yeah.
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 Well, I vote both are fine. You know, I'm also fine with signing the form and sending my edits and then just having him work with you, Andrew, to make
Okay.
sure it's implemented.
Okay. Yeah, that works. We'll take your signature as contingent upon completion of your comments. So,
Sounds good.
okay, cool. Well, thank you. I'm going to let Lloyd back in here.
Yeah.
You there, Lloyd?
I don't see him. Oh, there he goes.
Lloyd, congratulations.
Woo!
Yes.
Yeah.
Thumbs up invisibly.
Cool. Well, good job, Lloyd. As far as paperwork and timing of stuff and edits, we'll figure that out. We weren't exactly sure of the timing required of that, but we are prepared to sign and all that.
Well, yeah, so I think since technically the fall semester, I'll have December to get everything in, so I don't think we're up against any deadline, but I want to get all wrapped up sooner.
Okay, cool. And then we, the committee, will get you comments. I don't know if you have anybody's already, but we'll get you comments as soon as we can so that you can start working on incorporating those. But with regard to whatever signatures and whatnot, just let us know. We are prepared to sign those. So
Great.
cool. Well, congrats again. Good job. Very nice.
Can't wait for an in-person celebration.
Yeah. Yeah. All righty. Well, cool.
Oh wait, were you asking about an in-person celebration, Lloyd?
Well, I said I can't wait for one,
Yes, yes.
whatever that is.
We could do an outdoors one, like widely separated in a circle or something.